Good afternoon. So today we're going to learn a little bit about Class 1 MHC processing made by me. So to review, we know that MHC is found on the surface of all cells and it helps the immune system recognize foreign substances. As we know, MHC stands for Major Histocompatibility Complex, and it's also known as the human leukocyte antigen. There are two major types of MHC protein molecules, Class 1 and Class 2 MHC. For this particular video, we're only going to review Class 1 MHC processing. Class 1 MHC can be found in almost all cells in an organism, while Class 2 MHC is only found in like macrophages and lymphocytes. So I think MHC class 1 molecules are really cool because they can be found on all nucleated cells in the body, which includes the same cells where MHC class 2 can be found, which are like antigen-presenting cells, dendritic cells, macrophages, and B cells. But MHC class 1 is not found on non-nucleated cells, such as red blood cells. So as we see in the diagram below, MHC class 1 proteins consist of two polypeptide subunits, an alpha chain, also known as a heavy chain, and a beta 2 microglobulin. So as we see here, the alpha chain is formed and organized into three extracellular alpha domains. Alpha 1, which is right here, Alpha 2, and Alpha 3. It is also composed of a transmembrane region because it goes through the membrane. And it's composed of a cytoplasmic tail, as you can see right under here. So the beta 2 microglobulin chain is composed of beta-pleated sheets, and it interacts with all three domains of the alpha chain to provide molecular stability and appropriate affinity for specific peptides within the binding cleft. Okay, so now that we know the structure, we want to know what is the purpose of this molecule. And the purpose of this molecule is to alert cytotoxic T cells about intracellular activities. They accomplish this by binding small peptides and presenting them extracellularly. So the immune system uses different pathways to eliminate intracellular and extracellular antigens. As a general rule, endogenous antigens, which are those that are generated within the cell, they're processed in the cytosolic pathway and presented on the membrane with class 1 MHC. So what we're going to see today is how a nucleated cell goes and processes MHC class 1. But in order to process MHC class 1, a cell basically has to be infected. So what we have here, it's the membrane of our cell, and we all know it's a lipid bilayer. And there's the extracellular fluid and the intracellular fluid. We have the rough ER and a nucleus, TAP 1 and TAP 2, which are all used in this pathway. So what happens is that a pathogen on the outside, which can be an endogenous pathogen, a virus or a bacteria, the pathogen invades the cell and it's basically infecting the cell. So now we have the pathogen inside the cell and it makes its own proteins, causing this infection to be endogenous. So now we have these proteins right here. And some proteins have very short half-lives. They're either denatured, misfolded, or otherwise they're just abnormal proteins, and they are degraded rapidly. These intracellular proteins are degraded into short peptides by a cytosolic proteolytic system present in all cells, and it's called the proteasome. So it has a barrel-like structure, and many proteins that are targeted for proteolysis have a small protein called ubiquitin attached to them. And the degradation of the ubiquitin protein complex seems to happen in the center of the proteasome, causing it to break down and having produce small peptides for presentation with MHC class 1. Then these small peptides enter the endoplasmic reticulum using TAP1 and TAP2. And as we know, TAP1 and TAP2 stands for transporter associated with antigen processing. Both TAP1 and TAP2 belong to the family of ATP binding cassette proteins found in the membranes of many cells, including bacteria. These proteins mediate ATP-dependent transport of amino acids, sugars, ions, and peptides. So now we have these small peptides inside the endoplasmic reticulum, which can actually be further broken down in the ER by other enzymes. So in the rough ER, we know it's called rough because it has ribosomes on there, and ribosomes make proteins, which is where a lot of the structure of MHC comes from. So they have the MHC structure, but it's only composed of alpha 1, alpha 2, and alpha 3. The incomplete MHC class 1 is bound to a protein that we're going to see in just a minute called connexin. Connexin is basically a chaperone protein. And then we have this another protein, which is the beta-2 macroglobulin, that eventually attaches to the MHC. Once a peptide is binded to the MHC class 1 molecule, the chaperone proteins dissociate. And now this MHC class 1 molecule, along with the peptide, is ready to leave the rough ER. This MHC class 1 molecule, together with a peptide, will leave the rough ER using an endosome, and it will go through the Golgi apparatus and out through the cell membrane, expressing the MHC class 1 molecule with the peptide. So remember that the small peptide is a small fragment of the bacteria that had previously infected the cell. The MHC class 1 molecule and the peptide will wait for interaction with the CD8 T cell. So I've drawn this MHC class 1 molecule and its peptide a little bit bigger than it's supposed to be, but this is how MHC class 1 is processed and presented on an unnucleated cell. I hope you guys really liked the video, and thanks for putting up with my awful drawings.